Hello again. Let's talk a little bit about some of the functional qualities of the cell membrane. In particular, let's talk about how the cell membrane gets involved in regulating the transport of materials from the outside to the inside, from the inside to outside. Remember that membrane is an important barrier. Just to remind you of the structural features of the membrane, remember we said it was a fluid mosaic, phospholipid bilayer with proteins embedded in it of various types. And we said that it was a selectively permeable membrane. Now, passive diffusion can occur right through the membrane. Remember, passive diffusion is a downhill process. It's a spontaneous process. It's when a substance is moving from a region where it's more highly concentrated to a region where it's less highly concentrated. The movement of the substance is just due to its kinetic energy. And so some substances are just going to move through membranes, moving right in between the molecules of the membrane, okay, from a region of higher to lower concentration. Simple diffusion, passive process, okay? Relatively selective because only those substances that can fit between the molecules in the membrane or fit through the structure of the molecules are going to be able to move by this simple diffusion process. But for many substances, it's quite important, especially small non-charged particles move through this way. Most of the gases that you could think of, CO2, O2, nitrogen gas, N2, they're all going to move back and forth through the surface of the cell by this simple diffusion process. Now, also, we'd expect to find moving through the membrane water molecules. And remember, we said that the movement of a solvent through a membrane, the movement of water, the key solvent in living systems, is called osmosis. And so water can move back and forth through a membrane. And there's two different ways in which we could talk about how the water's moving. We could say that the water is moving from an area of lower solute concentration to an area of higher solute concentration. Now, you probably know from the other videos you've seen of mine that that's not really the way I prefer to talk about it. And instead, if I'm focusing on the water molecule moving, then what I'd say is that in osmosis, water is moving from or traveling primarily from an area of higher water solvent concentration to an area of lower water or solvent concentration. If you go back and forth between these two slides, what you'll see is that I'm talking about the same thing. It's just that in one, I'm referring to the two areas in terms of their solute concentration. In the other one, I'm talking about the two areas in relation to their solvent concentration. Remember that osmosis is a type of simple diffusion. It's just a passive diffusion of a water molecule downhill from regions of what? Higher water content to regions of lower water content. All right? Now, just to remind you also, another feature, that cells have to be very concerned about water regulation. We are aqueous organisms, aren't we? We have bags of aqueous solution. Our organelles are little bags of different aqueous solutions. But from the cell's point of view overall, regulating water flow is a very important feature for the organism. And we talked previously about how cells in isotonic solutions have a movement of water into and out of the cell that's equal, a net osmosis of 0. And in that case, we'd have plant cells that are in their normal condition, animal cells that are in their normal state, or plant cells that are in their flaccid state. If I place a cell into a solution that's hypotonic compared to the cell's interior, we'd expect the cell to gain more water than it loses, net intracellular osmosis, and it might lyse if it's an animal cell. It might become very stiff and turgid if it's a plant cell. And the reverse can also occur. If an animal or plant cell is placed into a solution that's hypertonic compared to the cell then has a net loss of water, a net extracellular osmosis, and we might expect to see an animal cell go through crenation or shriveling, and we might also see a plant cell shrivel, but in that case it's plasmolyzed or plasmolysis has occurred. All right, so this movement of water through a membrane is one of the very important features of what a cell is helping to regulate. All right, in the membrane of cells, however, there are plenty of proteins that are associated with regulating transport. So if I have a molecule that moves through a protein to get from one side of the membrane to another, and if that molecule is moving from high to low concentration, If it's a passive process, a downhill process, then we refer to it as facilitated diffusion. Notice it's still diffusion. It's still a type of passive downhill process, moving from higher to lower concentration. But now the substance is moving through special passageways, transport proteins, gateways, all right? Channels or carrier proteins that allow the substance to move through their structure to get from one side to another, all right? So this type of movement is called facilitated diffusion. Cells might also expend some of their own energy to make a substance move against its concentration gradient, to make a substance move uphill. Now, if the cell has to use some of its own stored energy, usually it's energy stored in the form of ATP, to cause a substance to move against its concentration gradient from low to high concentration, then we refer to that kind of process as active transport. And it also requires a protein be involved. So notice in this particular representation, we're seeing a molecule that's moving substances, well, in one case, it's moving a substance from the inside to the outside of the cell. And in the other case, we're moving a substance from the outside to the inside of the cell. This would be a kind of protein that's referred to as a carrier protein because it changes shape. It would also be referred to as a protein that's an antiport protein because it's moving substances in both directions. But let's go back to the idea of it being an active transport process. We're saying that for the substance to move out of the cell in this first condition, it requires that the protein that's moving it gain some energy from ATP to push it through the membrane. Likewise, in the second illustration, we're saying that the protein needs to go through another shape change, a change in its conformation, to pull the substance through the membrane. And notice that that's associated also with that phosphate group that had been added to the protein before by the ATP. So we're dealing with movement across a membrane by a transport protein. The transport protein has to change shape to make the movement take place, and it's ATP energy that's being utilized to move the substance. Notice ATP, hydrolysis, adding a phosphate group to the protein. Remember what we said that does. Changes the shape of the protein. The protein then, because of its shape change, releases the solute molecule to the other side of the membrane. If we consider the other aspect of this, now we're moving a molecule across the membrane in the other direction. The molecule binds to the protein. The release of the phosphate group from the protein now causes it to revert back to its original shape, and the substance is released on the other side. All right, so we're dealing with movement across a membrane that requires the input of energy, ATP energy in this case, to make the substance move. Now, what if I want to move more than just one solute at a time? What if I want to move more than just a couple molecules at a time? I want to move large things, or I want to move whole clusters of things. Well, now I'm going to have to carry out processes that are called exocytosis and endocytosis. And in both of these processes, it requires that we actually have membrane-like bags of material. Remember, we used the term vesicles to refer to these membrane-like bags of material that are found either within the cell or are brought into the cell from the outside. And so in this first illustration, we're looking at, for example, something like one of the vesicles that would have come off the Golgi apparatus. Remember the Golgi apparatus we talked about previously? So we would have something that would be an exocytotic vesicle. A vesicle that's been made by the Golgi has some kind of material we want to release to the outside environment. Well, notice what's happening in the illustration. The vesicle's membrane fuses with the cell membrane. When it fuses with the cell membrane, it opens, and the materials that were contained within the vesicle are released to the outside. Exo, outside, cytosis of the cell. So we talk about this as an exocytotic process. Well, what if I want to move a cluster of materials or a large material into the cell? As you might imagine, I'm going to be doing the reverse of that process, and therefore we call it endocytosis. In this particular case, notice how the membrane is creating a little bit of a pocket that materials collect in. It creates a depression on its surface, and materials collect there. That little depression, that little cluster of membrane molecules, phospholipids, pinch off to form an endocytotic vesicle, and we've brought something inside. By the way, this also could be the cell membrane stretching out around a bacterium as a cell tries to consume a foreign material. But in both cases, I'm going to end up with an endocytotic vesicle. And if you remember, we talked about how lysosomes then bind to these endocytotic vesicles and help in the processing and the digestion of the materials that were brought inside. So endocytosis is when I create a vesicle out of my plasma membrane to bring some type of material inside. It could be a cluster of small molecules. It also could be something as large as a bacterial cell. All right? Now, just for your purposes, realize that you're going to see that referred to, let me distinguish them again, that if I'm doing endocytosis and I'm bringing in a cluster of small molecules, you'll see that called penocytosis. It literally means cell drinking. If, on the other hand, I'm taking in a large structure or a whole bacterial cell with my membrane, then you'll see that sometimes referred to as a type of endocytosis called phagocytosis. And the prefix phago means to eat. So sometimes you'll see endocytosis subdivided into penocytosis and phagocytosis. Cell drinking, cell eating. Okay, so let's consider what else might happen in terms of the membrane. What if I want to bring things in, but I don't want to bring in sort of a general mix of stuff. I want to bring in a specific thing. What we find is that many cells have on their surface what are called receptor proteins. The receptor proteins have a particular shape. That shape will fit with or attach to a type of molecule called a ligand. The word ligand is a very general term. It means any molecule that binds to a receptor. And so in this particular case, notice what we're saying, that we have a particle, an LDL particle. LDL stands for low density lipoprotein. That particle has on its surface protein ligands. Those protein ligands can attach to receptor proteins in the surface of the cell. So now the cell, instead of just taking in a sort of a random mix of materials that are outside, it's going to be taking in just specifically these particular particles. So this type of endocytosis is referred to as receptor-mediated endocytosis, because we're bringing something inside the cell, inside of a membrane bag, but we're doing it because it has bound to specific receptors on the surface of the cell. So there's our three broad kinds of endocytosis that we've been talking about. I mentioned phagocytosis, when a cell surrounds and consumes some large particle of material or maybe another cell. And in this particular illustration, we see an amoeba wrapping itself around and creating a container around a food particle it's bringing in, phagocytosis. Penocytosis, when the membrane creates little pockets that the materials outside the cell fall into. And I drink in a mixture of materials along with the fluid outside the cell. And then finally, receptor-mediated endocytosis, where specific structures outside the cell, ligands, bind to specific receptors in the membrane of the cell and cluster together in one region so that the cell can bring in a number of that one particular molecule all at the same time. So, three forms of endocytosis: phagocytosis, penocytosis, and receptor-mediated endocytosis. Now, just to give you an idea of how these kinds of systems might malfunction, there's a disease called familial hypercholesterolemia. And what it basically means is that there sometimes are people that inherit an inability to bring cholesterol molecules into cells effectively. As a result, the cholesterol levels in their bloodstream are extremely high. Regardless of their diet, they have persistently high levels of cholesterol. When that was studied, it was found that the problem existed in this receptor-mediated system, that the people were making an incorrectly shaped receptor protein. As a result, they couldn't grab a hold of these LDL particles. The ligand on the particle wouldn't bind to the receptor, and therefore their cells weren't able to take in the LDL particles. Those people have very high levels of circulating cholesterol and unfortunately have a great deal of problems with heart disease. And there's nothing that they can really do in terms of changing their diet or anything, because our bodies normally make cholesterol and pass it around between cells. And the normal events that occur are to capture those cholesterol packages and pull them inside cells. If I can't do that, they have to stay in the extracellular environment, and unfortunately there's a number of negative side effects associated with that. All right, well then let's consider what we've been looking at. We've been focusing on the transport processes associated with the cell membrane and its function. We said that some substances can diffuse simply right through, right in between the molecules of the cell membrane. Now they have to be of a certain size, and they have to be able to pass through or blend with the molecules of the cell membrane. And so that happens for a number of different kinds of small things, especially gases like O2 and N2. We said that there could be simple diffusion of solvent molecules through the cell membrane, and we took a look at osmosis. Remember, also in a previous discussion, we talked about a solute molecule diffusing through the membrane, and we said that that would be referred to as dialysis, didn't we? Osmosis, though, is a type of simple diffusion, the movement of water molecules through the cell membrane from a region of higher to a region of lower concentration. We said some substances have to move through special protein passageways. They're still going to be moving by simple diffusion from high to low, but through a protein passageway, and therefore we would talk about that as a type of facilitated diffusion. Interestingly enough, for some cells, they have specialized proteins for letting water pass through. So notice osmosis may occur by simple diffusion for some cells, and it might occur by facilitated diffusion for other special kinds of cells. So both of these are passive processes. They're both types of diffusion, downhill processes. We contrasted that with active transport, which is an uphill process. We said the uphill process is one that requires an input of energy. The cell has to spend some of its stored energy to get it to take place. All right? Uphill. Finally, we talked about the various different forms of endocytosis, and in general, we focused on the idea that this was the membrane creating some kind of pocket to bring materials inside. And then we took a moment to focus on one example of receptor-mediated endocytosis, where the membrane had specific receptors to help particular molecules with ligands present move into the cell. All right, so you can see the cell is a very dynamic structure, regulating a number of activities. And we're going to see that as we look at other kinds of membranes in the cell, that a lot of these same kinds of processes occur with these other membranes. Don't think of this as being exclusively the cell or plasma membrane. It might be the membrane of the mitochondria. It might be part of the nuclear envelope. It might be part of the Golgi apparatus. It might be part of any of those membrane structures we talked about previously. All right, I hope you enjoyed this presentation. I look forward to talking to you the next time.